Buenas tardes a todas, a todos. Bienvenidos, bienvenidas a esta nueva charla de la serie de charlas relacionadas con el estudio de las cuencas hidrográficas latinoamericanas. Antes de empezar formalmente con la charla, quiero recordar y agradecer nuevamente a quienes han participado en la organización de esta serie de charlas. Por una parte, a la Universidad de Antioquia que nos ha colaborado con todo lo necesario para poder hacer estas transmisiones, a la Red Colombiana de Limnología y a Juan David por siempre estar presentes y por permitir transmitir esto a través del canal de YouTube y por mantenerlo como un repositorio de las charlas que hemos venido haciendo tanto este año como los años anteriores. Y me parece muy importante también agradecer al FAPESP y a la Universidad Federal de Sao Carlos, que a través de los convenios que se han hecho entre las universidades brasileras y la Universidad de Antioquia principalmente, pues nos ha permitido como llegar no solamente a la gente de la red µSudAqua, sino también a otros investigadores de Colombia y de Sudamérica. Y finalmente, pero no menos importante, a todos los miembros de la red µSudAqua por su participación, por su interés. Les recuerdo que una de las intenciones de esta charla es generar ideas para la reunión que vamos a tener el próximo octubre para discutir sobre de qué manera vamos a trabajar con los datos que se están generando como parte de la iniciativa Wonders. Brevemente voy a introducir a nuestro invitado en español, luego cambiaré a inglés. Nuestro invitado de hoy es James Tegen. James es un investigador reconocido internacionalmente en el área de la ecología teórica. Trabaja en la interfase entre la ecología microbiana, la hidrobiogeoquímica y en general sobre el estudio de las cuencas hídricas. En este momento es parte del Pacific Northwest National Laboratory y pues ha tenido estudios en biología en la Universidad Central de Washington. Su máster también lo hizo en la misma universidad y posteriormente hizo su doctorado en la Universidad de Arizona en ecología y evolución. Dentro de la hoja de vida de James, que es muy amplia, quiero resaltar un par de cosas que me parecen importantes. Por una parte, es un miembro muy activo de la comunidad científica de Norteamérica, es miembro de la Sociedad Ecológica de América, es miembro de la Sociedad de Ciencia del Agua y miembro de la Unión Geofísica Americana. Entonces está envuelto en muchos temas del estudio limnológico y ecológico de las aguas. James ha participado en muchas iniciativas, eh, entre otras cosas, ha sido eh, co-líder del Gordon Research Seminar on Metabolic Ecology, y ha estado invitado para participar en el Center for Ecological Analysis and Synthesis en un grupo de trabajo sobre beta diversidad. Y una de las principales razones para que lo hayamos invitado hoy es porque en este momento lleva varios años liderando una iniciativa internacional que se llama Wonders, Worldwide Hydrobiochemical Observation Network for Dynamics River Systems, que es un consorcio de investigación que busca como tratar de buscar los links entre la hidrología, la biogeoquímica y los procesos microbianos en los ríos, principalmente alrededor del mundo. Entonces, como parte de esta iniciativa, estamos trabajando desde hace ya un par de años para que desde Sudamérica nos podamos vincular a esta iniciativa, y pues gracias a James que nos facilitó su participación, estamos muestreando varias cuencas latinoamericanas. Entonces, en este momento voy a cambiar a inglés y vamos a darle la palabra a James. James, hello, how are you? Welcome here and thank you for accepting this invitation.
Hello. Yes, thank you for the introduction, Juan Pablo, and really appreciate the opportunity. Thanks everyone for joining. I think this is my first live stream YouTube presentation. That's quite fun. Glad that's happening. Okay.
Uh,
Oh,
so
let's see.
I was telling the people just to let you know that as part of the Wonders initiative that you are leading, we met a few years ago with the µSudAqua network, and thanks to that at that meeting, now we are starting to get the samples from different catchments in South America
Mhm.
as part of your initiative. And hopefully we will be able to participate and to collaborate and contribute from the South American perspective to this interesting project. So the stage is yours, and thank you for being here again.
Yes, wonderful. And I too, oh, let's see. Is it sharing? Is it good?
Yeah,
Yeah.
it looks very
Okay,
good. Thank you.
wonderful. Okay. Yes, we too, in terms of the collaboration you were just mentioning and the sampling that's happening, definitely one of the main goals of that is to provide opportunities, new opportunities for us to be collaborating together. Um, and so I very much look forward to that. So today's presentation is not going to touch on that work. It's going to focus on thinking about organic matter chemistry through the lens of community ecology. And as was mentioned in Juan Pablo's introduction, I'm at Pacific Northwest National Laboratory. That's up in the northwest part of the continental United States. And this slide deck is a Google slide deck. And if I provided the link early in the chat, Juan Pablo, if you can help, that'd be great to share it again. Let me know if anyone has trouble accessing it. Encourage you certainly to reach out with any questions. Okay, first, what I wanted to do is start with a bit of a motivation of why we're gonna talk about organic matter chemistry in rivers and streams and why. Why are we even studying rivers and streams? And so the way we think about this is that the Earth's terrestrial landscape, and if this is the United States here, that landscape is organized fundamentally into watersheds. And those watersheds are drained, of course, by streams and rivers that are sometimes thought of as like the veins of the Earth. They take material from the landscape, from hill slopes, etc. They move into streams and rivers, and they transport that material downstream. And as that's happening, of course, there's a lot of transformations that are going on. A lot of greenhouse gases are released, for example, CO2 is produced, methane, etc. And historically, these streams and rivers were thought of as passive pipes, just transporting material downstream into the marine environment. And in 2007, there's this famous paper by Cole et al. That really clearly rejected that idea that these are neutral pipes just transmitting material, transporting material, and that rivers and streams, and more broadly, freshwater ecosystems are a vital part of the global carbon cycle and really do deserve our attention, as they state. More recently, last year, Baton et al. Provided a similar kind of perspective in this quote. As you can see, river networks are the largest biogeochemical nexus between continents, oceans, and the atmosphere on the planet. And again, emphasizing the important role that these systems play, and really outsized role. They cover a very, very small percentage of the Earth's land surface, but contribute in terms of net fluxes of CO2 of similar magnitudes as the ocean and soil and more terrestrial systems. So a really important part. And we can think about, okay, what is it within these systems that's so important? And one thing that we think about is if you imagine yourself standing on the edge of a river or a stream, you see the water flowing by, and underneath that are the sediments, the material you're standing on if you're ever going around wading in the river. We can think about those sediments are often a major biogeochemical reactor in these systems. We think of this as riverbed sediment respiration. Sometimes we call it ER-SED. And this is something of great interest to us from an ecosystem perspective. And why? Like, why do we care about sediment respiration? And there are several reasons. One is that it can contribute up to 100% of the respiration, the metabolism that's going on in a given stream reach. And in the map on the right, this is some of our recent data. This is in the Yakima River Basin. This is in the northwest part of the US, up here in Washington State. It's about 16,000 square kilometers in area to give you a sense of scale. And all the dots on here are places where we estimated field reach, stream reach scale respiration rates. And the purple colors are places where the sediments contribute almost all of that respiration, meaning the reactions in the water column aren't contributing much at all. But it's interesting. So that means, okay, the sediments are quite important. However, there's also yellow dots on that map, yellow locations. Those are places where the sediments actually aren't that important. Most of the reactions are happening in the water column, which is the second bullet there, where it says sediment respiration it's important. It's also variable. It can be down to even 3% of whole stream respiration. But there's no model that can really predict that at this point. So this is highly uncertain in terms of how important are the reactions in sediments and how does that vary? And part of the reason that's uncertain is because it's relatively understudied. There's a lot of data sets in the field looking at whole stream respiration, but relatively few that actually examine and parse out the contributions from sediments in the water column. And so that helps motivate us of getting into starting to try to understand the mechanisms that govern respiration rates in sediments. And that then leads us down this path of going towards organic matter chemistry. And why do we do that? Well, one reason, some work from one of our team members, Vanessa Garrido Caruso, pictured here. This is a paper from a few years ago. And this is a laboratory incubation now. So this is lab scale, not the field scale stuff I was just showing you. And the key point of this figure, and there's a link there to that to the paper. If you can open the slides, that should be a live link in the lower left there if you want to see the paper. What the data are showing you is the vertical axis is respiration rate. So basically going up the vertical axis is more respiration, increasing flux, increasing rate. On the horizontal axis is organic matter thermodynamic favorability. Basically, as you go to the left on the horizontal axis, the organic matter chemistry is increasingly favorable for microbial respiration, meaning microbes get more energy from organic matter as you move to the left. So then there's this relationship. Basically, you're getting increasing sediment respiration when you have increasingly favorable organic matter chemistry. And we've seen that in other studies. There's another example of this. This is Aditi Sengupta's study. I can show you a live link if you want to go look at it. Same kinds of axes. Sorry, I flipped the arrow. It meant to change that. The vertical axis, again, increasing flux going up. Horizontal axis, organic matter favorability. It's the same relationship. It's decreasing favorability going to the right, meaning increasing favorability going to the left. Same relationship, separate study. We've seen this also in modeling studies. We've seen this in field scale studies. We keep seeing it coming up. And it's suggesting to us, okay, there's something quite important about these thermodynamic components of organic matter chemistry and just more generally organic matter chemistry. And then thinking about, okay, how should we be thinking about organic matter chemistry? How should we try to understand that? And today we're really talking about just one lens, one way of looking at it through this community ecology perspective. And certainly there are other ways. And one reason that inspired us to start to think about looking at organic matter chemistry through a lens of community ecology is seeing relationships like this. So what we have here is, again, from Aditi's paper, same vertical axis. This is respiration rate in the lab. On the horizontal axis now has to do with the microbial community. So not the chemistry yet, just the microbial community. And what you're seeing is that as deterministic ecological selection becomes stronger, moving to the right, respiration rates are actually getting slower. So there's this link between how communities are put together from an ecological perspective and this biogeochemical rate. And so that got us thinking of like, okay, if there's this connection between community assembly and biogeochemistry and these important systems, can we think about organic matter chemistry through a similar ecological lens of deterministic organization of communities, but think about molecules instead of microbes? And that then leads to this paper that we put together, led by Bob Danzak. I think Bob is here today. You're going to see him show up several times in this presentation. He's been really a key innovator for linking community ecology to organic matter chemistry. Again, this should be a live link. So let's step through and think about an ecological lens. I keep saying this, like community ecology. What do I mean by it in this case? So if you think about an ecological community, how is it put together? Like, what are the high level general processes that organize a community? And that's kind of shown it in the table here. So the middle column, that's organisms. So the process, basically you have birth, which is reproduction. You have organisms that die, okay, you have death, and you have movement. Those are the three high level general processes that lead to any ecological community. Can we think about then molecules through the same conceptual perspective? And I think that we can. This is what Bob's paper lays out, is that we can think about molecules in the same way. Any sample of organic matter is the result of birth in terms of production of organic molecules. Death is kind of like transformation. It's changed in some way. Well, now it's a new molecule. And movement in the sense of molecules passively moving, diffusing, being transported around. So we started to think about, okay, there's actually these nice conceptual parallels. And then we can start to think about when we think about microbial communities, we think about deterministic selection and stochasticity. So let's talk about deterministic selection. What does that mean? That means that from an ecological, biological sense, a community is deterministically assembled when taxa are very specifically selected for or against going across conditions. So for example, moving across a pH gradient, that's going to select for and against certain kinds of microbes. That's a deterministic filter, a selective process. Here again, we can extend that idea into organic molecules in the sense that a composition and assemblage of organic molecules can be put together deterministically if certain molecules are systematically preferentially produced or transformed across some environmental condition. The complement to determinism is stochasticity. And in this case, thinking about, okay, how does that influence an ecological community, a microbial community, for example? In this case, you have stochastic variability when the rate of dispersal of individuals either restricts where taxa can be, if it's very, very limited and they can't move, that can actually lead to stochastic assembly. Or the same taxa are everywhere because dispersal is very, very fast. That can also lead to stochasticity. That's from an ecological, like microbial perspective. We can extend that idea then into molecules, organic molecules. The transport of molecules can either restrict where certain molecules are, if there's a very, very limited transport, or take a stream that's really well mixed. It's moving everything around. That's going to cause stochastic assembly of organic molecules into an assemblage that you would observe in any given sample. So the point here is just laying out these ideas that there are these very strong conceptual parallels between how we can view ecological communities and how we can view organic matter assemblages of organic molecules. I think it definitely point you to Bob's paper that lays out that whole theoretical construct and does a bunch of analyses, and there's scripts and everything. So then let's start to ask, okay, how are we actually going to study assembly processes, determinism and stochasticity in organic molecules? How are we going to do that? Well, we want an untargeted way to characterize those samples. So we're going to use what's called FTICR mass spectrometry. Some of you may be familiar with it. I'm not going to go in the real details of this today. The short story is that it's untargeted, very high resolution method that can observe thousands of organic molecules in any given sample. And basically, it gives you data that you can kind of see on the right there, where you have lots of peaks that show up at different masses. And that's what comes out of a mass spectrometer. And the resolution is so high in terms of the mass resolution, you can actually infer the molecular formula of those peaks. So there's in blue, there's an example. You can see an example molecular formula. So this is going to give us a way in any given sample, lots of formulas, and that's what we're going to end up studying to think about stochasticity and determinism. And then how do we do that? So this is a figure. I think this one's from Bob's 2020 paper that I linked before. So what Bob did is he created a method to take these molecular formulas and turn them into a dendrogram. And I'll show you how we actually characterize assembly processes. A key thing that we need in order to infer the level of determinism and stochasticity, we need to have a phylogeny in the case of if we're talking about microbial communities, and we need something like that, but for molecules, if we're studying organic matter chemistry. So we call that a dendrogram. And in this case, this is the molecular characteristics dendrogram, or MCD. So what Bob did is he took all these formulas you get out of the mass spectrometer, and then you say, okay, that's actually a representation of their chemistry. You can look at stoichiometric ratios, you can look at the mass of those peaks and several other derived properties. And it's basically functional traits of the organic molecules. And from that, you can then build the dendrogram based off of relationships among those various kinds of traits. You can see a zoom in. That's one that should be one sample set that's shown there in that dendrogram on the lower left, and a zoom in. So every tip is a formula, and that's what's shown in the middle. So what we're going to do is once we have that, so here's the primary steps. We've assigned formula from the samples and whatever sample set we're studying. We've defined some characteristics, as I was saying, things like stoichiometric ratios and some other derived properties I'm not going to go into details about. Then we build this dendrogram, like this functional trait relationships, basically. We calculate the branch lengths then that separate different molecules, and we can compare those branch lengths to a stochastic expectation. So let's walk through what that looks like. And first, I'm just going to show you, well, just visualize it on a dendrogram, and I'll go through a little bit more of the method. So here what we have is we have a similar dendrogram that I was just showing you. And let's think about what it looks like when you have a dendrogram and organic molecules are stochastically assembled into assemblages into a given sample. This is what it looks like. So the colors, if you imagine two samples, you went to an upstream location and you went to some other river in some coastal area. And so we get these two samples. And then let's think about which molecules, really which tips of the dendrogram are shared. That's in yellow. The ones that are unique to sample one are in blue. The ones that are unique to sample two are in red. And this is what it looks like when there's stochastic assembly. It's just everything's mixed up. There's no real patterns. There's no real structure, as you'd expect when it's stochastic. But what does it look like in reality? That's now on the right-hand side. This is what the patterns look like when you just take the observed dendrogram and you put the shared and unique formula onto the dendrogram. The point here is that there is a lot of structure, a lot of organization that is not stochastic when we look at the real dendrogram, at least in some parts of it. But in other parts, you can see like in the lower, kind of the lower half of that dendrogram, it's kind of just random and it looks similar to the stochastic expectation. So the title of this slide says, okay, organic matter has both deterministic and stochastic components, just like ecological communities. So the point here is hopefully giving you a sense that this is a reasonable thing to be doing and thinking that we do have deterministic and stochastic components. So then let's then go through a little bit more how do we actually then go through and quantify stochasticity and determinism? This is an old figure that was set up for microbial communities, but it applies just as well to the organic molecules in those dendrograms. And how this works is, so in this case, it's community one, community two. You could replace that with sample one and sample two. So what you have is you have the dendrogram. That's the branches there kind of on the left. And you have your observed two samples. And say you just have um, two molecules in each sample. Those are going to be the solid circles for the first one and open circles for the second community or assemblage. So that's in red. That's the observed. And what you do is you then calculate, okay, how far apart are those molecules from each other on this dendrogram? What is the branch length between them? And you quantify that using this metric called beta MNTD. I'm not going to get in the fine details of exactly what that is, but that's the observation. How far apart are they on the dendrogram? And then you take the location of those molecules on the dendrogram, and then you randomize it. You just mix them up. That's in blue. And then you recalculate the branch lengths, separating community one from community two, or assemblage one from assemblage two. You recalculate it. And then you do that randomization basically 1,000 times. And that's what gives you that blue beta MNTD distribution on the right-hand side there. Now what you do is you take your observation, the value in red that you just have one number for, and you compare it to that whole distribution. And basically the difference between the mean of the blue, the peak of the blue, and where the red is, that's beta NTI, beta nearest taxon index. And that's measured in units of standard deviation. How many standard deviations is the red from the peak of the blue? And you can do that for all kinds of pairwise comparisons. Now, if you think about comparing across 100 samples, every time it's a pairwise comparison. And so then you can get a distribution of beta NTI by doing this process for every single pairwise comparison. That's why there's a distribution shown there on the right. And basically, the further away beta NTI is from zero, the further you move either way, towards the positive or the negative, the further you move, the stronger deterministic selection is. And what we're going to see today is mostly data out on the right-hand side above the +2 significance threshold. +2 and -2 are significance thresholds because it's measured in standard deviations. So if you're above 2 standard deviations or less than 2 standard deviations on the other side, it's significant, indicates significant deterministic selection. This has been done many times in microbial communities and tree communities and bird communities and things like that. And what we've been doing is applying this whole approach to organic matter now. Okay, now, oh boy, that really advanced. Now, we're going to take that whole approach. All we've been talking about is methods now. We're going to apply it to some data sets. The first data set I'm going to show you it comes from dry parts of stream beds, sometimes called a parafluvial zone or a non-perennial stream. Just very briefly, why are we studying organic matter chemistry in dry stream bed sediments? The reason is that most streams go dry once a year. µSudAqua showed that in 2021 in Nature on the left. Also, streams are getting drier. That's shown by Zipper et al. In 2021 there. So stream bed sediments drying out and re-wetting are the most common kind of sediments on the planet. And so we want to study organic matter chemistry in those in particular. So what did we do to do that? We were able to generate samples. I'm sorry, hold on just a second. Having technical struggle. Okay, so the way that we did this is we generated samples from dry regions of streams across the US. And so you can see the yellow dots here. There's some up in Alaska, up in the upper left. You have the continental US, and then a couple from Puerto Rico as well, the yellow dots. And I want to emphasize, how did we actually get those samples? That was done by working with the Wonders Consortium. I'm going to show you that in just a second if it lets me. Okay, there we go. So Juan Pablo mentioned Wonders in the introductory remarks, and I just want to talk through a little bit about what Wonders is before we go into some of the results. So Wonders is this consortium. Um, there's the name, is founded in 2017. There's a website there, and it's a participatory science network. The goal is generating data, knowledge, models, opportunities consistently across the world's rivers and researchers that are interested in those systems. And one of the key things about Wonders is how we do science. It's just as important as what we do, we feel. So as I said, it's a global consortium. It's for and by the scientific community. Hydrology, biogeochemistry, microbiology. We try to generate resources that are useful to teams, individuals beyond our core team. So we're trying to accelerate river corridor science that's transferable across very diverse rivers and also to achieve mutual benefit. So this is what we're doing right now is having this conversation, this presentation is trying to reach out, connect the collaboration that we've built with several of you in terms of generating samples is also in that spirit of trying to find mutual benefit. And we do that by using what are called icon science principles. So icon science is about being intentionally integrated, coordinated, open, and networked. I'll define those in just a moment. But it's really, this is a framework that applies across all domains of science. Several publications there you could look into. It's really about purposefully striving for mutual benefit and knowledge that is transferable across very diverse systems. So those four words, integrated, coordinated, open, and networked, that's how Wonders works. We base ourselves off these principles. And it's being integrated across disciplines to encourage diverse insights. It's coordinated with very consistent protocols to enable transferability and interoperability. We do our best to be open throughout the whole research lifecycle. We're trying to promote knowledge exchange and engagement. We're networked, where we're trying to design and implement our work with a broad range of interested parties, such as yourselves. That is to facilitate mutual benefit, as I mentioned. You can take a look at a couple. We've started the Icon Science Cooperative. I'm not going to go into the details today. There's a website there. Would love to engage with y'all in this space. So start to bring it back now. So again, we're trying to get samples from lots of places to do the organic matter chemistry, that null modeling, the beta NTI with the Wonders Consortium. So basically, we're sending out sampling kits that are all consistent. They have biology, chemistry, physical properties from that integrated perspective. These kits were designed in collaboration with the science community, not just our team, again, trying to do icon science. The kits go out. They don't cost people anything. We send them out very consistent. There's a protocol in video form and written form. Interested people reach out, we send it to them, they go to the field, they follow the protocols, they collect very consistent metadata that's represented in the lower left. The samples come back to us and we do all the processing and the data generation. We have very diverse sediments that come back. That's represented in that picture on the lower bottom there. Really, I love that picture. It's like a rainbow of sediments that come back from these efforts. And our team then processes those. That's Dilman Delgado there processing some of the samples. So this is how we're getting samples through Wonders, applying according to ICON principles. Let's come back though. Let's come back to the technical stuff. So in this particular study, we're getting samples through Wonders from all those yellow dots that are shown there. We're using FTICR analysis on the organic matter from those samples, sediment-associated organic matter. That's the picture in the upper right. And we're generating the dendrogram, doing the null modeling, getting beta NTI from all those samples. Some details. So at every one of those yellow dots, the sampling design is there are 10 samples in a given stream reach. So imagine you're at a stream, looking upstream, downstream, 10 different locations going up and down the stream. So that happens at every one of the yellow dots. And then what we do is we build one dendrogram for every site. So 10 samples, one site, one dendrogram. Then we calculate that beta NTI for all of the 45 pairwise comparisons. So when you go from 10 samples, if I did the math right, 10 samples gives you 45 unique pairwise comparisons if you compare all samples to each other. And then we can ask the question, okay, is organic matter organized deterministically with very high beta NTI values? Is it stochastic? What does it look like? So here's some results. This is a paper that's just coming into press now. And across all those sites, I'll emphasize the purple line. That is the distribution of beta NTI values across all those samples, all those pairwise comparisons. And the red dashed lines, those are the significance thresholds, +2, -2. So the horizontal axis is beta NTI. It's the same purple line on both the panels here. Short story is most organic matter chemistry in most sites is deterministically organized. There's a little bit of stochasticity. If you're in between the dashed red lines, that's stochastic. If you're to the right, that's deterministic. It's mostly deterministic. These two distributions, the blue bars, are two different sites, two yellow dots. So what I'm showing here is the least deterministic site or the most stochastic. That's on the left. So there is some sites that are quite stochastic, actually, and other sites that are very, very deterministic. So that's just kind of hitting the two ends of the whole range. Here's another way of looking at the data. So now these blue bars, the blue distribution, the histogram, is taking for every yellow dot, we get a median beta NTI. So you get the 45 comparisons, you get the median value, and then you make a histogram out of it. And what you can see, again, it's mostly deterministic. It's on the right-hand side of that red dashed line, which means it's that's something called variable selection, where processes are driving organic matter chemistry to be very different as you go from sample to sample to sample. There's deterministic divergence in the composition, even though we're sampling very similar sediment within each site. And we can compare that to a certain degree. We can compare that to some previous data. Those are two of Bob's previous papers. And you can see you have data from surface water, stream water, and pore water. So if you go into the sediments and you just pull some water out when you're in the field, that would be pore water in this case. So you can see there's some stochasticity, there's some determinism. We're in the general range that we've seen in previous studies. It's kind of a main point there. So we can start to ask, okay, what's explaining variation in the level of determinism? And we started to try to investigate that in this particular study. So I mentioned that we're studying dry sediments. So you go to the field and you get some sediments where there's no water over top. But how much water is in sediments is actually still quite variable as you go across different sites. So what we have here is on the left-hand side, the vertical axis is the site level median beta NTI. The horizontal is a representation of sediment moisture. So on the left, that's how much water there is per gram of dry sediment. And then the regression in the black dots are the maximum values in that scatter plot. So basically doing a constraint boundary. And you can see this negative relationship. What that's saying is that the maximum strength of determinism, that is the highest value beta NTI, that goes down as moisture goes up. So if you have a lot of water relative to how much sediment you have more stochasticity. To say it the other way, if you have sediment where there's very, very little water and a lot of sediment, you're likely to be in a very deterministic situation for the organic matter chemistry. But what we found on the right-hand side is that depends on how you characterize water in sediments. If you normalize the water to how much dry sediment there is, you get the left-hand plot. If you just look at the percentage of water in a given sample, that's the right-hand panel, you get no relationship. That leads us to believe that there's something going on with organo-mineral interactions with the sediments themselves. There's kind of a deep story in there, but this is just to say there's some kind of link between moisture content and the level of determinism in organic matter. Okay. And then we started to try to put this together into a conceptual model. And from pulling from a couple of Bob's papers, I showed these data before, or part of these data before. We started to think about, okay, there's something about water, which might have to do also with movement, dispersal, transport of organic matter. And something that his papers showed is that if you look at surface water that's flowing down the stream and you compare it, the level of beta NTI to what's happening in the pore water, in both these studies, surface water has smaller values of beta NTI. It's more stochastic than the pore water. And so that started to help us think like, okay, how can we put all this together into a conceptual model and just propose it really as a hypothesis? This is the last part of the paper that is this paper that's in press, this conceptual model. And I should mention a lot of this particular paper was done very heavily with Vanessa Garbaru-Caruso that I mentioned before. And of course, Bob's in the mix with this one as well. The idea here, and this is really a hypothesis now, is that if you think about the level of determinism in stream water or river water, we're proposing that that's going to often be relatively stochastic. And as you move from that stream water into pore water, you'll have a little bit more determinism. And as you move further into those dry sediments, kind of moving up the bank, you'll be even more determinism. And as you move further up into the soil systems that like never inundate with water, they'll be the most deterministic part of a given, really watershed ecosystem. That's yet to be tested. I hope can inspire someone to go and actually test that idea. So we're just trying to put pieces of data together to come up with that at the very end of the paper. Okay, now there's a couple other things I'm going to go over. I'm looking at the clock. I'll try to close up in like maybe 5 minutes or so. So I'll maybe kind of go through these next ones with a little bit more speed. So then we can ask the question, let's focus on stream water. And if we go across streams, everything we've been talking about is within a given site. We can ask the question, is organic matter usually stochastic? Is it always stochastic? How does that vary? This is another Bob and Vanessa team here. This is the Yakima River Basin again. You can see in the northwest part of the US, a bunch of different sampling sites, surface water sampling, FTICR, same kind of thing we already talked about. One thing I'll mention is if you think about just diversity, so not the stochasticity stuff, determinism stuff, just diversity in terms of number of unique formulas. Think of it as like number of unique species, if you want to think about it from an ecological side. That goes up as you move down this particular watershed or basin, if you like, and you're sampling water as you move down the basin, the organic matter is getting more and more diverse. The greater number of unique formulas. Just kind of keep that in mind. Same thing now. Samples, FTICR, null modeling, beta NTI. What does that look like? So first of all, if you compare each sample, I should mention stream water. What is stream water? As you move down a watershed, you go from little tiny streams. Those are going to be like stream water 1 and then 2. And as you get bigger and bigger, stream water goes up and up. So down the watershed, higher stream water. If you compare stream water 2 across different to the other stream orders and compare stream order 3 to the other stream orders, and you look at beta NTI, this is what you get. There's no real pattern. It's all about the same. But what's super interesting, I think, is that if you look, and this is Bob did this null modeling. I take no credit for, for these results. These are the really cool stuff that Bob did here. So if you look within a stream order, so just stream order 2 compared to stream order 2, et cetera, you get this unimodal pattern. What it suggests is that when you're halfway down this watershed and you're comparing the streams that are all halfway down the watershed, that's where determinism is maximized. And then it decreases as you go to the bigger and bigger streams and rivers, you lose determinism and you get more stochastic. I was really surprised that it came out this way, but then Vanessa and Bob put this conceptual model together to try to think about this. So you think about a watershed, streams are connected as you're moving down, and you have relatively weak variable selection or deterministic selection in the upper small streams. As you're moving down into that kind of pink magenta part, That's where variable selection is maximized. And the idea, there's several possible ideas here. There's more diverse land cover, land use in the middle part of watersheds. This is where flow starts to slow down and residence time of the water goes up. And so you can have more processing going on as well. And then as you move further down the watershed to the left part of this diagram, you're just getting a lot of mixing. And that can then give you this more stochastic signal. So this particular paper is in review. Okay, so I think I have, yes, just two more slides. We'll end up pretty close to on time. So I think there's a lot more questions. We're just kind of barely scratching the surface here. Here's the two conceptual models that I've shown you, two hypotheses really that I think deserve further evaluation. There's lots more questions that we can ask. Bob also developed an approach where you can go beyond this sample level analysis. Everything we've talked about is beta NTI at the whole sample level. He developed a method, I didn't talk about it today, but he developed a method where you can do the null modeling on every single molecule. You get an answer for every molecule. Super cool. You can then connect it directly to individual microbial species, which we did in a paper that's just coming out in Nature Microbiology, actually in permafrost systems. You can start to look at scale dependencies. So a nice thing about this null modeling approach is that your answer that you get is actually dependent on the scale that you study the system. As you change scales, your answer changes. That's actually powerful because then you can start to ask at what scale do you move from stochasticity to determinism or determinism to stochasticity, etc. Further links to biogeochemistry. I started out with connecting to rates, respiration rates, a lot of opportunity to connect over into biogeochemical fluxes, looking at responses to disturbance. We have a PhD student with us right now. Today was his last day. Bob's been working with him, looking at how organic matter responds to disturbance in the Amazon using this null modeling approach. Okay, and then this is the last slide. There's a lot of opportunities. I've been talking a lot about the chemistry and the theory and null modeling and stuff. I want to back out, emphasize again our interest in doing science with lots of different people, providing opportunities, finding mutual benefit. So we can collaborate with pretty much anyone in the world in all kinds of different ways. We can give virtual presentations like this. We can do it within classrooms, within professional networks like this. We can partner on proposals, including internationally. We can have students visit us. I just mentioned the PhD student. He happens to be from Germany. We can host international students through various mechanisms. All of our data is open. It's all very well structured, very well documented. I think Brienne Forbes is on the call. She spends a lot of time managing those data to make sure they're useful for everybody. We can engage in a variety of different collaborations. We can do technical trainings. Bob's put together trainings for FTICR data analysis before. We can provide sampling kits as we are doing with some of you already. And with that, there's a QR code that'll take you to a Google form that Brienne Forbes put together. There's a variety of options of ways to collaborate with us. You can select some options and let us know if you want to engage in new and different ways. So I encourage you to reach out. So I think we have like 10 minutes or so. If there's any questions, Juan Pablo, I'll let you take it back over.
Thank you very much, James. So I think it was very nice to see your presentation. As you mentioned it, I think it's going to be very inspiring for some of us that we will be meeting on Buenos Aires next October. So for sure, we will use the information of your presentation. Very nice stuff going on. So let me see if there are a few questions. Thank James for this very interesting talk. Do you think, James, that the relative contribution of environmental factors versus the intrinsic composition of organic matter in shaping its degradation would vary depending on the biome or climate of river watersheds? Let's suppose that we are comparing similar sized watersheds. I can't hear you. Okay.
Yeah, sorry, I was muted. I was just thinking for a second. So if
Uh,
I think the answer is yes. So, or if I'm understanding the question, so there's going to be some influence of the intrinsic chemistry. So we can think about that in terms of the thermodynamic properties. That will influence how microbes are interacting with that chemistry. So that's like the intrinsic chemistry. But then there are a lot of other properties, um, for example, mineralogy of the sediments that have a really strong influence on how organic molecules are binding onto those mineral surfaces that can change their availability to microbial communities. So that can fundamentally change whether microbes can even access certain kinds of molecules. Another component is I talked about dry sediments. So the wetting and drying regime, that varies a lot, right? As you go across different biomes, more arid sorts of places, more tropical, wet places. So that wetting and drying regime will strongly, strongly influence degradation patterns, these stochasticity and deterministic kind of lenses on those, in part through the either the inhibition or facilitation of aggregates. Soil aggregates can develop when you have more dry conditions. So you have roots, so you get plants, roots into the soil. Those really promote aggregation of particles together. That can strongly protect organic matter from microbial degradation as well. So there's a combination of physical and biological and chemical at play. So anyways, maybe I'm saying too much, but a lot of interactions that will vary significantly moving across different biomes and watersheds, even of the same size.
Okay, thank you. So you were talking about the link between microbial component and the chemistry of the organic matter. So uh, following your conceptual framework, do you think that the deterministic molecules are going to vary with the deterministic part of the community, and what's going on with the stochastic part of the organic matter? Also, I was wondering, maybe I missed it, but what proportion of the molecules are stochastic and what proportion are deterministic?
Okay, love this. I know this wasn't expected. So Bob Danzek is here. Bob, do you want to try? Are you interested in answering this question? If not, I certainly can try.
I can try.
Just wanted to also give Bob a chance for you to meet Bob and, kind of put a face there
Thank you,
as well.
Bob.
Hey, I still have the same background. But yeah, so whether or not deterministic microbes are linked to deterministic compounds, I think is going to be very ecosystem kind of dependent in a way, because I can imagine scenarios where photodegradation might cause the organic matter to shift in a deterministic way that isn't necessarily reflected in the microbial community, at least one to one. I mean, I expect there to be a pass down effect of some sort, but not necessarily one to one. Because we did do some. We looked into this briefly, not in as much detail um, as we would have liked with like transcriptomic data or something like that. But we did look at um, microbial community feature level kind of determinism compared to organic matter feature level determinism, and related the two to find possible areas of uh, coincidence. And there were some interesting patterns. Nothing I would say were super landmark. It could have been a data set thing. It could be any of those things, but um, I don't have a good, short answer is I don't have a good sense of whether or not they'd be directly connected. And I think that's an area of exciting exploration, especially with greater omics resolution. I think that's as we get more functional understanding of microbes, I think some of those connections will start to bear out better.
Okay, no, perfect. Thank you very much. It's going to be very interesting to explore this new information. Thank you, Bob. So, Vicky, I think you want to ask a question. Are you
Yeah.
there?
Yeah, do you hear me?
Yes, sure.
Okay, great. Thank you, James. It's always nice to hear you. It was a wonderful talk. Just because I already read the paper from the transect, right? I would think that the less moisture samples in the transect, the ones that are more stochastic, like sorry, more deterministic, variable selection. Yeah.
Yep.
So that is a more selective environment for organic matter. But yeah, I have the same question as Juan Pablo. Is that a more deterministic environment for bacteria as well? Because I'm thinking about it. If in the water, in the mainstream, you can have different inputs of organic matter, right? Because you have terrestrial input and also planktonic input from phytoplankton and different things. So it's like the origin of the organic matter can vary more in the water stream, right?
Sure. Yeah, I think so. Yeah.
So you have different sources, more variable sources in this in the water than in the drier sediments. So that should modify the active bacteria living there, because you have different sources availability for organic matter to consume, right?
Sure. Yes. And I agree with you. And we've conceptualized this as when you measure organic matter in the surface water, you're really measuring an integrated signal of what's going on in the upstream watershed, which is basically, I think, what you're saying. All these different sources of organic matter when you measure in the surface water. It's very integrative. And when you look into the sediments, you're really looking at processes happening in that kind of sample volume. It's much, much smaller, far more constrained environment with less diverse range of organic matter sources. And I think that's one reason that we're getting these very deterministic signals. And one reason that, at least when we've done the comparisons, the surface water shows a little bit more stochasticity because there's more mixing going on from these various sources. I think one thing you also asked is, would the microbes be more deterministic in the dry sediments? And I'm sure the answer isn't always yes, but one experiment we did, it was that Aditi Sengupta paper that's linked. In that paper, we see exactly that. So when the sediments are dry, the microbial community becomes much more deterministic. However, we only see it when we look at the ribosomal RNA, the expressed RNA. We do not see it with the genomic DNA, which makes sense, right? There's not enough time for the DNA, the whole community to change, but who's active totally changes. It's this big deterministic signal that comes up.
Yeah, also I think respiration rate, it talks about activity, but you're not distinguishing which molecular formulas are being consumed. It's
True.
like a general activity, right?
In that particular paper, that is right. Yes, that's right. In other papers, where we've linked the thermodynamics to respiration rates. In those cases, we're making a stronger case for which molecules are actually being consumed and interacting. There's another paper led by, oh my gosh, now I've just forgotten the first author. That's terrible. This is more of a machine learning method where they identified regions of chemical space that appear to be bioavailable and are linked to respiration rates. Fernaz is his first name. I'm forgetting his last name. I'm so sorry.
Yeah, so the size of the molecule may be involved in their deterministic or stochastic processes, right?
I think so, yes. The size and the thermodynamic efficiency as well. And yes, and there's some aspects of stoichiometry in the mix as well.
So thank you. And thank you, Bob. It was nice to see you.
Okay, great. So perhaps we can finish with a last general question, maybe from, I guess, from people, someone working on limnology. They say, very interesting topic. Would you define, I'm translating, so would you define what you do as limnochemistry, or how do you define it?
So very interesting. We generally think of what we do as hydrobiogeochemistry. With almost everything we do has a strong hydrology context that it's set within. So then we've got the biology, we've got the chemistry, physical, chemical, biological, all put together. So that's our integrative term that we normally use as a high level.
Okay, perfect. So thank you, James, again. I think it was very interesting and very productive. Hopefully, this is not the last time that we meet, and we can hear your information and what you have to tell. And thank you, everyone, for being here. Gracias. We finish now. There is another. Do you think? Do you have time for another question, James, or?
Yes, and I think I'm gonna do the similar thing that I did before,
Okay.
and see if Vanessa Garaburu Caruso would like to answer this one. I think, is it the
Okay.
one in the chat?
Yeah, that one. Thank you.
Okay. Yeah, Vanessa,
Thanks,
do you want to
Vanessa.
try?
Yes.
Hi, everyone. Nice to see you. Sorry I was a little late.
No problem.
I think, part of the things that we are observing in that study that James mentioned from the Yakima River Basin can suggest that water residence time would play a role. I don't know with the microbes because we don't have microbial data from that, but with the organic matter chemistry, that seems to be a potential factor. For that particular study, we sample only during base flow conditions. So we were not expecting a lot of rainfall events or many disturbances that could have had an effect on the water. But certainly other events like that could have an influence as well in the processes that we're observing. And thinking about disturbances and changes in water, we did another wonder study back in 2018 that we call the 48-hour study. And that one, we sampled 8 different rivers across all over the world where they had changes in water discharge every 2 hours or every so on that were driven by different mechanisms. And Bob, actually, I think he has a preprint out where he explores some null modeling on that data from these rivers that had changes in discharge. Bob, I don't know if you want to say a little more about that paper as well related to the question.
Okay, I think it's okay for now. So thank you, Vanessa, for your explanation. Thank you, Bob and James. Hopefully we meet very soon.
Yes, thanks so much. Really appreciate the opportunity. Great to see you all. Take
Okay,
care.
have a good afternoon, everyone.
Bye.
Hasta
Thank
luego
you.
a todos.